Hello, everyone. I'm Julie Haas. I'm one of the program committee members, and I'm here with Danielle Bassett, who spoke this morning on controlling the brain. And she's here to answer a couple of questions. We have a couple of questions that have already been posted, but of course, you're free to pose your own. So maybe should we go from the top down?
Yeah, sounds good.
Sounds good. Maybe I have some of these. I don't hear an echo, Thomas. Do you, Daniel?
I do not know.
Um, uh, so these were some questions that were posed earlier, so they're not from me. So one of the questions was, thanks for the great talk. Very interesting topic and great findings. I'm still curious about what the role nonlinearities, both in the dynamics and the application of control, might play, especially in the context of state changes. I'd expect that we're no longer in a close linearizing point around the original state, such that a linear approximation might die in there.
Yeah, that's a good question. I think that we need, this raises the question of how far you move with specific state changes and whether that movement can still be estimated with the linearization of a nonlinear model. And I think that the answer depends a little bit on the way that you have defined the state. So maybe I will bring that up as a rich place to think about this. So when we're thinking about fMRI data, for example, from human volunteers, if we think of each TR as a state, and we move from one TR to the next TR, so one sample to the next sample, then there is some evidence that that time scale can be well approximated by linear models. And some of that work has been done by other labs, and we've done some as well. So that, if you make the time step small enough and you make the estimates of the activities large enough, then you can do well in the linear approximation. But if you go down, obviously, to very small spatial scales, so talking about single neurons spiking, obviously the nonlinearity becomes highly important. We do have a paper that is on arXiv now and under review, which kind of provides some factors that are relevant for different imaging modalities, from EEG to fMRI to spiking neurons, where that can tell you whether the linear model is going to be roughly good or not particularly good. So I'll try to pull that up and stick it in the chat here. But it's a great question, and I think it really does depend on your definition of state.
Okay. Let's see, I think so a next question we have is from a previous question. If it's understood correctly, the main point is to measure the control energy needed for state transitions. But the questioner wants to know, why did you then include the distance between states, so adjacency matrix, into your cost function?
Yeah, that's a good question, because the distance between states is not just dependent on the activation profiles. You could take two states of activation and calculate the L2 norm as like a simple, Euclidean distance, but that doesn't take into account how the activity will flow and what the constraints are of the pipes. So that's why we take into account the adjacency matrix, the structure, because that allows us to be more realistic about how activity may be propagating in the system and how that forms a constraint on how far away states are. So that's why the adjacency matrix comes in.
Okay, great. So a next question is I have another question from the previous session. How could the control energy or signals be related to any markers or brain stimulation strategies from clinical applications? In the natural systems, should we limit ourselves to restricting control signals only to sensory systems? So that, the entry points we have to the brain, or?
Yeah, that's a really good question. I think in terms of the entry states or entry regions, so where you would actually be injecting control, I think that if you are attempting to model the brain's response to a cognitive task where visual stimuli are coming in or auditory stimuli are coming in, then using auditory or visual regions as the points where the control signal is being injected makes a lot of sense. It's consistent with what's happening in the task. However, when it comes to brain stimulation for clinical populations, we know that stimulating regions that are not sensory becomes very relevant. So, for example, in the treatment of Parkinson's disease, that is not a sensory area that's being stimulated. So I think the answer of where the energy is being injected depends on what you are trying to do. Are you trying to model what happens in a cognitively demanding task, or are you trying to stimulate a region for clinical purposes to assist in the writing of dynamics?
Mm-hmm.
I guess the first part of the question, which is how can this be related to brain stimulation strategies? We do have a paper the first author is Jennifer Stiso. And in that paper, it was published in Cell Reports in 2019, we study the use of electrical grid stimulation in patients who have medically refractory epilepsy. And so we say, well, here we know what the structural connectivity is from the white matter, and we know which regions have stimulation being injected into them. Let's use our control model and say, if they stimulated this region, where should the brain go? And is our prediction from our model consistent with what we actually see as the brain moves after stimulation? And Jenny sees a really nice statistical similarity between our predictions and what actually happens. There's still, however, a lot of variance left unexplained. So it is a relatively simple model. So there's more to do here in digging down into how to best predict the response of the brain to stimulation. But there's at least preliminary suggestions that this model provides significant variance explained.
Mm-hmm. So you mentioned this, if I can follow up, you mentioned the stimulation that we use for Parkinson's disease. Has your work pointed towards any different areas that we might not have thought yet to use, inhibiting amygdala, for instance, or something similar?
That's a really good question. It's not a space that my lab has really tackled over the last few years. We've been more focused on modeling cognitive tasks instead. But I do think that that would be a really interesting direction to take the work.
Okay. We have a question from Thomas, who I'll invite on screen. Thomas.
Hello.
Hello.
Hi, Danielle. Hi, Julie. Yeah, I was wondering, in cases where you have individual connectomes, connectivity, how much variability do you see actually in these control energies, or also in the other type of energy?
Yeah, that's a good question. So we do see, I guess I could pull up my slides, but in the last study where we are evaluating working memory performance, we do see significant variability. I had the box plots and then I had all of the individual data points, and there's significant variation there, which also is correlated with individual differences on the accuracy in their performance of the task. So that means that there is that, and that variation is partially coming from differences in the structural connectivity. So that indicates to us that the individual variability certainly matters. And similarly, in the study of development, where I showed that the control energy was correlated with executive performance on a computerized neurocognitive battery, all of that variability comes just from the white matter because the states themselves are fixed. So it's a zero state and then an activation of frontal parietal regions with like a binary activation. And so all of that variation comes from the structure. So those two examples indicate to us that there's significant variability in white matter that is not only coming out in our estimates of control energy, but also explanatory of behavior in these two contexts.
Okay, I see. I was about to follow up to say, yeah, is this kind of correlated with the behavior, and therefore it's something that's very meaningful to how people can do the tasks, or is it just something where some brain is wired up more strongly or less strongly? It doesn't make so much difference for the task. But I think you have already answered that, so thank you very much.
Yeah. Thanks. Good question.
If I can follow up on something from that. So I remember that your work showed that controllability increased with age and seemed to asymptote towards 20 or so.
Mm-hmm.
Do you? I mean, that implies that that's the developmental process of maturation of a human. Is that what you think it is? I mean, do you think we can voluntarily increase that after 20 or?
That's a great question. We have not used these techniques to study training paradigms that have been linked, for example, to changes in white matter. So I know that there are some really nice work from Heidi Johansenberg and colleagues showing that training on specific motor tasks and other labs showing training on language, learning new languages, can drive changes in the white matter. We have not used those sorts of paradigms yet to evaluate whether changes in the white matter that are driven by really intensive training as adults could lead to a change in the energy landscape on which the brain is moving. I think that that's the natural hypothesis, though, from our work and also from your question. So I think that would be a neat direction, but it's not one we've taken yet.
Yeah, so much work to do. So I think the last question we have, unless we have more questions from all the folks in the audience, are there quantitative estimates of how the control energy cost relates to metabolic energy usage in the network by the brain? Is there a simple relationship?
Yeah, this is a good question, and it's similar to an earlier one. And I had mentioned at the end of the main talk that we have some data in the pipeline showing that the control energy that we estimate is related to metabolic estimates of energy consumption using PET imaging. So connecting up our control energy estimates with estimates from PET imaging for the metabolism. And that is something that we're hoping will be on a preprint server this summer. But it's a nice initial evidence that there is a relationship between our theoretically estimated control energy and energies that are actually used in the brain. This is at the large scale, though, and I think there's still a lot to do both at the large scale in populations where we see metabolic changes, for example, in schizophrenia, but also in at the smaller scale of single neurons or small groups of neurons, where some of these measurements can be done more precisely. So I think that's an important route to follow up on.
Yeah, and maybe some bold signaling in there would be an interesting thing to combine.
Yeah, absolutely. Or cerebral blood flow too.
Mm-hmm.
Yeah.
Yeah. All right. So I'd like to leave us open for a few questions from our audience at the point. I'm almost out of my own questions here, or pre-canned questions. And since this isn't a class that we're teaching, we won't pick on people. Okay, I guess that's all the questions we have, really. Oh, there's another one that popped up. All right, maybe. So Jessica Mullick, can I invite you on screen? There is one. All right.
Can you hear me?
Yeah. Great.
I really enjoyed your talk. Thank you so much. So I was interested in control energy in the case of a clinical disorder that progresses in different stages, such as substance use or Parkinson's disease. Such disorders may also include phasic changes, like, for example, a state of drug craving or withdrawal, or you can even think of something like stress that might not be in the context of a disorder. But there's also progression through different phases of the disorder, like heavy use or recovery. Do you think there might be differences between the more phasic versus more sustained changes in these kind of disorders?
Yes, that's a really great question, and it gives me an opportunity to highlight some of the work by my colleague, Julie Blendy, and her graduate student, Kate Brynildson, that I just put in the chat window. So what they were interested in doing is studying mice who have opiates, who use opiates and become dependent and then are taken through withdrawal, or the drug is removed and then they go through withdrawal. And so what they did is that they study activation profiles in these in across the stages of dependence and withdrawal, and then also study which regions of the brain are have the most control capacity in each of these different stages. In that study, they use the connectome that from the mouse that is available from the Allen Brain Institute. So this is the first study that uses the network control theory approach with substance use, and in this more kind of progressive fashion. So I think it's a neat first evaluation. I think again, there's more to do here. It would be wonderful to complement the structural connectivity estimates from the Allen Brain Institute with an effective connectivity measurement, and also to take the animals perhaps into a to have bold measurements as well in a high intensity small animal magnet. So I think there's exciting directions to take that work, but the approach itself is flexible in its applicability in that context.
Thank you so much. Very interesting.
We have one more question from Jeremy Oka. So nothing is happening when I click invite user on screen here, so I'm not sure we're going to get Jeremy on screen. But he's asking what I think is a really interesting question. Are the controlling nodes always controlling, or do they come in and out of controlling mode?
Yeah, that's a really, really good question. What we find is that for some state transitions, one set of regions will be the optimal controllers, and then for a different state transition, another set of regions will be the optimal controllers. So that's one broad way in which different regions can be the controllers, depending on where the brain needs to go, in some sense. But even beyond that, you can ask Jeremy's question over time, and you can ask in the actual trajectory, is the controlling node that is important early in the trajectory the same nodes that are important later in the trajectory? And the answer is that they can be quite different. So initially you may want to have this set of regions push you in one direction, and then you have another set of regions that pull you over to the final state. So in that way, there can be regions that are important for early in the strategy, or early in the trajectory versus later in the trajectory. We also find, and there's a paper I'll try to pull up from Evelyn Tang, that some regions are really structurally predisposed to enact persistent control, so a control that where energy isn't being injected for a long period of time, and then other regions are very structurally predisposed to be able to enact transient control. Like I can send a signal and it will make a big impact quickly, and then it will dampen out really quickly based on the way I'm structurally connected to the rest of the network. So that's interesting, and something that I would love to study further is why are certain regions more structurally predisposed for this transient versus the persistent control, and does the brain use them in those ways to kind of flexibly modulate the dynamics that are being enacted at any given moment.
And what's the range of timescales over which those switches happen, or variations?
Yeah, in the context of fMRI, we are studying pretty long timescales, right? It's on the scale of tens to hundreds of seconds. But on the scale, at the cellular scale, it could be quite different. That's just not a place where we've done a lot of the work.
Jeremy has a follow-up for you. How are maps of controllers and controlled nodes connected to resting state networks? And whether the resting state networks are only some kind of epiphenomenon deducible from this controllability measure?
That's an interesting question. So one thing that you can do is, to answer your question, Jeremy, is to take the brain and say, if the brain was allowed to move to any state, and it was allowed to start at any state and end at any state, what state is going to be the easiest to move to from any starting point? So imagine there's random starting points, and you ask, where would the brain naturally go to as a focal target state that would take the least amount of energy from anywhere it could have begun? And that low energy state, or the state that requires lowest energy to get to, is one that has strong activation of areas in the precuneus and superior frontal areas, and areas that are known to be very active in the default mode of the brain. And so that leads us to, question, is it possible that the reason that the brain moves in this default mode state of activation of these particular areas is because that is the lowest energy place to exist, given this pattern of structural connectivity that the brain has? So I would say that is an interesting network control explanation for the location of the default mode regions. But I think Jeremy's question more broadly about other circuits is open, and I don't think that that's something that we have a good answer to yet.
Okay, so that's it. So Thomas has a follow-up to that, and I'll get to Evie's question in a second. Thomas.
Hey, me again. Sorry, I didn't want to monopolize the discussion, but this just occurred to me, and I'm not even sure it's a sensible question, but I'll ask it anyway and then show my innocence here. You talked about different regions being relevant as controllers along a trajectory to a desired brain state just a couple of questions ago. And I was wondering, is there a sensible understanding, and what's your speculation, who's orchestrating this dynamic? So how does the brain do it so that the right regions take control to get in the most efficient way to the desired state?
Yeah, that's such a good question. And I think it highlights something that I've been thinking about a lot in how to expand the current model that we have. So an important limitation of the current model is that it treats all regions as equivalent. So it's based on one adjacency matrix and activity flows to each region along the white matter and through that region according to how white matter comes out of it, right? So every region is the same. You can change that by having local estimates of neurotransmitter levels, and that's a paper that I had mentioned earlier. I'll try to find the bioarchives link for that one from another lab at Cornell. But it still doesn't, so that would change what happens when the activity comes in and how it flows out, in the sense that it would change the magnitude, but it wouldn't change the nature of the activity, the nature of the information. So there is no explicit way of having computation in a region in this particular model. And I think what is really exciting and hard, but intriguing is how can we use this model and add in computations at single regions, so that then we allow for a flow of activity, a calculation occurring there, and then a flow of activity after that. If you allow computations at single regions, then you don't get an orchestrator necessarily, but you get a lot more capacity to, oh, well, this one is definitely going to spread activity and this one's not, because this is a calculation of an AND gate or an OR gate or whatever it happens to be, right? So I think that's the natural progression for this model in terms of allowing for more sophisticated decisions to happen at single regions.
Right, otherwise everything would be predetermined by the connectivity. We would be just doing, maybe to some extent we are, right? We're just doing what our connectivity tells us to do, right?
Yeah, well, and by the fact that the brain is not a closed system. It's so open. It's open to the rest of the body. It's open to the world through our eyes and our ears and our tactile sensations and all sorts of things. So there is incoming stimuli all the time. There's always energy being injected in the sense of our model. And so that would explain very rich dynamics, but and probably not really deterministic dynamics, given the non-deterministic nature of our world in some sense. But I do think that there needs to be, to bridge the network control explanation of cognitive control and executive function with beautiful computational models of what cognitive control does in gray matter. I think we need to put these two things together. I think they provide two complementary explanations. The gray matter does a calculation, but the white matter transmits the activity after that calculation to other regions. So both are true, and in neither model are they combined. So I would love to put the two together, because I think that's where we will have a richer understanding.
Cool, thank you very much.
And we have a mentoring question from Evie, who I'll also invite on screen. Although nothing is happening when I'm clicking invite. Oh, something's happening. Real-time report of internet speed. All right, there's Evie.
Hello.
Hello.
I was wondering, can you recommend something to students who are currently starting, as undergraduates, and given your trajectory from Cavendish Lab, straight physics into neuroscience? So what helped you, sustain your interest in what you're doing, and what skills, what areas were valuable in developing your area of inquiry?
Yeah, that's a good question. I mean, I really like basic training in math and physics, just because it does become very relevant later for all kinds of questions, and it provides you with a lot of flexibility in the kinds of questions you want to ask later in your career. So I've found that to be, I feel lucky and happy that was something that I chose early. I guess I would also say, however, that physicists have sometimes have a negative reputation for coming into fields and assuming that they can provide, the answer to a very important question without really understanding the details of the measurements, the types of questions that field has been working on, and all of the domain knowledge. This is not something that is just a reputation of physicists, but also happens in many other fields, where someone comes in and says, but I have this great idea, I can solve it for you. And I guess I would say that there's, to the undergrad who wants to work in an interdisciplinary space, I think I would say just do as much as you can to maintain a humility in understanding that there is so much that's known in each of the fields that you may attempt to contribute to. And so to bring with your excitement and passion, bring some humility too, because I think that that can help to lead you to important scientific discoveries in collaboration and communication with the other fields involved. So one of the ways that I try to engage in interdisciplinary work is also to pull in experts from various fields. If I feel that we are not knowledgeable enough in one particular vein, I say, well, let's bring in somebody and have them be part of this collaboration, have us really think about it from their perspective too. And I found that in general, that makes the science so much better and so much more exciting. So yeah, I don't know, that's what I would say.
Thank you very much.
So what do you, if I can follow up, what do you feel like the straightest path is to figuring out the domain knowledge level kind of questions that you should be asking? Is that a specified question?
Yeah, that is. I mean, sometimes it is asking multiple people questions about, I think that this area would be relevant. Can you tell me more about it? If we tried to use it this way, does that make sense? Does it not make sense? What are the assumptions? And sometimes it takes several conversations with different people, different experts in a field to recognize kind of what level of explanation you are hoping to provide and what level of explanation a technique or a field is able to give. And there's somehow a matching that has to happen between those two. And I don't think it's really obvious necessarily from the outset. It is sometimes a fairly circuitous path to find where there's a match.
Yeah, I feel like that's the hard part. And so, yeah, figuring out how to navigate that.
Yeah.
Okay. Well, once again, I'd like to open it up to any more questions from the attendees here. If we don't have any other questions, then I'll thank you for a truly invigorating talk.
Thank you so much for having me and for all of the great questions.
Thank you. All right. Nice to have you.
Thank you.
All right, and I think I will log off. I'm not supposed to end the broadcast, but I will log off here and thank everyone for coming.